Okay, so my talk's a little bit different in that today I'm really going to talk more about why people get cancer, the current existing biomarkers of cancer risk, which I and probably many of the clinical oncologists will say are kind of limited. MicroRNAs is potentially new and exciting biomarkers of cancer risk. I hope I can convince you of that today. And one such example of that in the KRAS gene, which we're referring to as the KRAS variant. This work's all been done in collaboration with Frank Slack. We have many ongoing projects, and as a disclosure, he and I are both founders of a company that's licensed this IP. Okay, so why do people get cancer? And all of us could probably name a couple of these cancer syndromes. This is why people get cancer. However, you know, these are inherited germline mutations in these important genes, many of which manage DNA and prevent a lot of DNA repair. And people that inherit these mutations usually inherit one bum copy, and then over life, their tumor loses the other copy, and they develop tumors. These are very rare, and these are usually pretty early onset cancers. So that is not why the majority of people get cancer. Instead, it does tell us, however, that it probably is in the DNA, you know, why people get cancer, not just in their resulting tumor cells, but there's probably a chance that things that go wrong in your DNA maintenance through life could put you at an increased risk for cancer. So, you know, what we see is really that most people develop cancer as they age. It's considered a disease of people aging. I won't use the word elderly, but 3/4 of people are over 55 when they're diagnosed with cancer. As I'm approaching that eventually too, so don't shoot me down. But it's true, most people get cancer as they age, and is it just because you're getting older? The truth is not everyone gets cancer, so it isn't the same for everyone. And who in this room, when they hit 55, is going to get cancer? You know, people are. It isn't that we're all not. And I'll tell you, as an oncologist, every cancer patient I have, they're surprised they have cancer. No one knows they're going to get cancer. So I think it's important, and I think we have a real problem right now in helping know who's going to get cancer. Right now, at the most, 5 to 10% of all cancers worldwide can be attributed to a known cause, a genetic cause. And again, many of those are these early onset childhood cancers, very strong familial history. But what about the rest of us? There's really a lot of room for discovery. And it's important, I think, because particularly when it's starting to be questioned, should people be screened even with useful screening tools? You know, mammograms not until you're 50. It's just a bummer if you get your breast cancer before you're 50. But the truth is it's not very cost effective to screen everyone when only a subset of people will get cancer. The problem is we don't have a way to identify those people. But it really could matter. You could direct screening programs, do appropriate screening. There could be lifestyle changes. Some cancers can be prevented, ovarian cancer possibly. And maybe you could direct future therapy. If you really know, hey, this group of cancers is caused by this, and we can address it this way. So I'm going to talk about microRNAs now, and in a very basic way introduce them. And I think microRNAs are our road to discovering a lot more markers of cancer risk. And I'm going to present one that we found today. So, you know, the paradigm that we always had, this blue thing is supposed to be a cell, but from your DNA, you made a messenger RNA, which made protein, and that kind of was the whole story. Until about 10 years ago, and this was an enormous body of excellent work in literature. In fact, though, there is a whole other class of RNA species that comes from the DNA. They're called microRNAs. They never turn into protein, so much for that RNA to DNA thing. DNA to RNA to protein thing. Instead, their job is just to be the boss of the messenger RNAs. And what's interesting about microRNAs is they're really master controllers. So they're really the tip of the iceberg or the top of the pyramid. So a single microRNA regulates hundreds of messenger RNAs. And it's important very critical processes. Development, they are in charge of your full development. Differentiation, stemness, cell death, the stress response, and aging. MicroRNAs orchestrate all of that. So you can imagine very easily how if you lose control of any of these things by disruption of a microRNA, you're going to get cancer. And in fact, it's really a lot of literature, very convincing that microRNAs are abnormal in every cancer type. If you look at a cancer and you look at the normal tissue, microRNAs are at significantly different levels. So why is that? And is it possible that these disruptions are the first thing that happens that causes cancer? So one other part I just want to explain a little more fully because it's really relevant to the talk is how a microRNA works. I just want to point out that microRNAs bind to a region of the messenger RNA. I told you they bind messenger RNAs. It's this 3' poly-A tail, this 3' untranslated region. So the protein part of the messenger RNA is up here. This region, no one ever thought it was important. Really, it hasn't been very well studied because before microRNAs, no one knew anything important bound there. So it's a very understudied region of the DNA and the messenger RNAs. We now know it's really important, and the discovery I'll talk about is found in just that very region. So I'm going to talk now about this collaboration between Frank and I and this discovery of a variant in this region of the DNA in the KRAS oncogene that is a biomarker for cancer risk. Okay, so Frank and I joined forces to study our two favorite things. Mine is the KRAS oncogene, probably the most important human cancer gene. It's abnormal in half of human cancers. It's associated with extreme resistance to treatment, which has always been of interest of mine. And the LET7 microRNA, again, probably the most important human microRNA dysregulated in cancer. Frank had shown earlier that LET7 and KRAS were LET7 regulated KRAS. We worked together to show that this was really important in protecting cells from radiation and stresses. So we came together and we said, you know what, let's try to figure out this relationship. Let's look in cancer patients, and let's look at this 3' untranslated region where no one's ever looked, and let's look where LET7 binds and see what we find. Sounds like a fishing expedition, but there really was some thoughts behind it. And I just want to point out here that LET7 binds a lot of times. Different microRNAs bind, but we focus on these regions. And we took the really, hard work approach. We sequenced this region. We didn't just say, all right, let's look at the databases and see if there's polymorphisms predicted. Let's look where microRNAs might bind. We actually sequenced this whole region, and we found quite a bit that hadn't been reported. But the important and interesting thing we found was a polymorphism, a change in one microRNA binding site. Now, you might say it's one microRNA binding site out of 10. But what was interesting is that it really was right in the middle of the LET7 microRNA binding site. And it was really common in our lung cancer patients. It was in 20% of them. It had never been reported, so that's a lot of lung cancer patients. It turns out it was germline. So if we had a patient that had a lung cancer and then a pancreatic cancer and a head and neck cancer all separate, they all had that same change in their DNA. This is in their germline DNA, in their spit, in their whole body DNA. And it was always in the normal tissue. So it's an inherited change in KRAS. This is in the KRAS gene where an important microRNA binds. We thought that was intriguing, and we went on. We have gone on, and this has been published. We showed that just the single change does change how LET7 binds, and it does change the levels of KRAS. So it's a functional polymorphism. We, in collaboration with Ken Kidd here at Yale, and you'll see a lot of in collaboration was, I love that about Yale. So much of the studies I'm presenting are group work from a lot of people here at Yale. But Ken has this fabulous, incredible collection of human DNA. This represents, it's 2,500 people representing 46 geographic populations. I bet I myself couldn't have named more than 5 geographic populations. But in general, in pink, those are Caucasian populations of European descent. There's African, there's Asian, there's Native American. You can see the polymorphism in general is not very common. Now, again, we found it in 20% of lung cancer patients. It's in 6% of people in the world, which is really in 3% of chromosomes, and in about 10 to 15% of Caucasian people, which were primarily the lung cancer patients we were studying. The point I just want to make here is that this really qualifies. It's in 7% of chromosomes for people that are into GWAS studies and genetics. This really qualifies as a rare variant and explains actually why this marker hasn't been picked up on some of those SNP GWAS studies, because they really don't see rare variants very well. And I can talk about that later if anyone wants. But we have gone on and evaluated this marker in many cancers now. And I want to summarize, and then I'm going to go on to what we think is our most exciting finding about this marker in ovarian cancer. But the first publication was in lung cancer. And in fact, that prevalence of 20% of our lung cancer patients was significant in 2 case controls. Just the presence of this single change in your DNA made you twice as likely to develop lung cancer. So that's amazing. That's just the average smoker, average bear. If you have this marker in this audience and you smoke, you are twice as likely to get lung cancer. So that's been published. Another group subsequently tested head and neck cancer. While they didn't find it was a risk for developing head and neck cancer, they found the 15% or so of people with head and neck cancer did significantly worse, with much worse survival. And that's been published as well. These are all ongoing studies and collaborations with people at Yale, a pancreatic case control with Harvey Risch and Herbert Yu, a uterine study with the same group, a breast study with Dave Tuck, Lindsay Harris, colon, that's an international study, prostate study here with Kevin Kelly. In all of these cancers, the marker does not appear to be a risk of getting the cancer, but in each case, it does seem to really significantly impact your response to therapy. Colon, for example, this is by another group, has shown that the presence of the KRAS variant predicts altered response to EGFR targeting agents. Now, we know KRAS mutations do that, but that's the first actually inherited KRAS change that also will predict response to therapy. So we're excited about that. And in these other cancers, it seems strongly associated with very aggressive subtypes of these cancers. So it might be important. Prostate cancer, you have all these people. All right, if you have the KRAS variant, you're much more likely to be hormone resistant and progress. And maybe we need to know that, and maybe it will help us decide how to treat you. Anyways, but overall, we've tested lots and lots of people, over 15,000 people. And in all these groups, it's in 15%, maybe 18% in lung, but not more in any group except for ovarian cancer. So now I'm going to tell you about that. So as a little background on ovarian cancer, I don't expect people to know a lot about it. It's not a particularly common cancer. It's 3% of cancers in women. That's 22,000 cases a year in the US. But it is a very deadly cancer. It's the leading cause of female GYN cancer-related death. And 15,000 people die just in the US every year of ovarian cancer. It is greatly feared by women because of a few things. One, there are no known risk factors besides being Caucasian and upper to middle class with fewer children. That's all. That's the only known risk factor. And people are essentially asymptomatic, and it strikes really at about 60 years of age. There's no disruption. You're postmenopausal. People don't notice you feel like you're gaining a little weight. Probably happens to a lot of people when they hit 60. I'm sure I'll feel the same way. I'm feeling like that at 40. But so people are scared of it, and it's very deadly. And the mainstays of treatment are really radical surgery and continual chemotherapy until it just overtakes your abdomen and you ultimately die of strangulation. So it's a really, really, really tough cancer. Now, there is some genetic risks associated with ovarian cancer, BRCA1 and 2 gene mutations, but they are really, really rare. They're often associated with early onset ovarian cancer, which isn't common. And this is based on family history, which I think we all know as treating oncologists is pretty limited. I mean, if you were adopted or your parents died at 50 or your grandparents died at 50, which wasn't that bad, you may not know if you're at risk for a cancer that happens at 60. So many other people might have a genetic risk for this cancer, since there are other not really known lifestyle risk factors. So we're hopeful that we have one. Okay, so we looked at our KRAS variant. I already told you what it was. It's never in more than 18% of any other group we've ever looked. And tested people at Yale, and we were very surprised to find that it was really enriched in ovarian cancer. So it was in almost a third of ovarian cancer patients. So this was not expected. So much so that we actually found people with other collections of ovarian cancer patients to confirm our results, one in collaboration with Dr. Santine here as well as with Herbert Yu. Now, 2 separate hospitals in Italy, again, just prospectively collected, not selected in any way. And again, it was really highly enriched in these ovarian cancer populations. So we said, hey, that sounds interesting, and maybe it's a marker of risk. It's at least a quarter of ovarian cancer patients. Versus at least at Yale in our controls, it was only in 10% of the controls. So it's not like everyone coming to Yale New Haven Hospital has this marker for some reason. That's just a depiction of that finding. We did go on to do now 2 case controls, and this paper has just been accepted to PNAS. But in 2 separate case controls, we were able to show that the presence of the marker, for a woman tested in this room, the presence of the marker tells you you are twice as likely to develop ovarian cancer. And there really are not markers that are out there that could be applied to general people without other familial risk factors or things. So, this is our Yale case control we constructed. These people are obviously matched for age, race, other risk factors, everything we could come up with. And this was supplied by Harvey Risch, the Connecticut case control. There's no overlap in these populations. We see the same findings. So we're very excited about that. But, we figured, all right, we're saying this is a risk for ovarian cancer. Let's really test it. Now, BRCA1 and 2, every other genetic risk factors, those were tested on high-risk populations. If we don't find our marker in high-risk populations, then there's something funny, and it can't be real. So we went to, there is this ovarian cancer syndrome called hereditary breast and ovarian cancer. And in collaboration with the genetics department here, we said, let's test these high-risk people. If our marker's real, it's going to be in these patients, and it's going to be in their families. So these were the patients. We started with people diagnosed with ovarian cancer. So something's going on. They have ovarian cancer, but they also have a family history that's documented by genetics, consistent with someone else with ovarian or breast. You know, some HBOC families are defined as 3 cases of ovarian cancer. Ours were not. It was a little more loosely defined, but there were some documented cases. Some of these patients themselves had both breast and ovarian cancer. So they were real, something was going on. They all were tested for the known markers of genetic risk, BRCA1 and 2, which in this type of group, historically, BRCA is associated with about half of them. And the rest, while there might be something going on, they're told they're uninformative. We don't know what your marker is. We have nothing to test your children or your family members. They are all considered high risk and you should be followed. And we tested our marker in these patients. So we started with the group that were BRCA negative. They have something going on, and we wanted to know if they had our marker. And we have 25 of these so far. The numbers continue to increase. But we were very, very pleased to find that our marker was in 60% of these people. So that is very strongly statistically significant. No one in the world has that, you know, no group in the world has a prevalence of 60%. So we're really, really excited about this. And, you know, for this really will predict an increase. It's just not a 2-fold increased risk. It's about a 6-fold increased risk for someone with this family history that tests positive for this marker. And it also would, for someone that has a mother that had the marker and they don't have the marker, they actually will predict they're at decreased risk of developing ovarian cancer. And that can be almost more valuable for some of these people that previously were just told you're uninformative and you should be followed. We then went on to the BRCA patients, and I'm going to tell you about this. This is ongoing work. We just think it's kind of interesting. You know, there are definitely these BRCA syndrome patients. And you know, we had about 33 that we also tested. We didn't know if we'd see it at 10% like the general population or 30% like the ovarian cancer population, but we saw something kind of interesting. Overall, it was in between. It was in 20% of these patients. However, what we saw was it was significantly more common to be in patients with breast or ovarian cancer with BRCA1 mutations. It's in 30% of them. And it was significantly less common to be in people with BRCA2 mutations, in less than 10% of them. So this was in our own cohort and then a collaboration with a group in the Netherlands in breast cancer patients. That's a dark blue. And you can see these look almost exactly the same. So we think there really is something here, and this is something we're, you know, actively pursuing now, trying to understand how the variant could modify in the background of BRCA mutations. So just briefly, I'm just going to show you an example of the type of family. So we now have many families, and we now have the KRAS variant. They have this family history of cancer. They themselves don't have any other gene mutations. This is just one family. So the square is a man, the circle is a woman, the slash means they've died. Black within their circle means they had a cancer. So you can see this is just one family. The grandmother and her sister had uterine breast cancer. Out of her 8 children, 5 of them had cancer. So that's a lot of cancer in a family. It's not early onset cancer. It's in their 70s, 60s, 70s, but they had cancer. You'll note 2 of them are lung cancer. Not a surprise. We showed our markers that predicts lung cancer risk. But then, you have fallopian, ovarian, a uterine. And then here's this next generation, which they're all told you're probably at increased risk, but we've had nothing to test you for. And they probably haven't developed their cancers yet because it seems to be a later onset cancer. Okay, so I'm going to briefly, in the last few minutes, just tell you a little bit about how people with ovarian cancer with the marker compare to people with ovarian cancer without. One important point is they are not significantly younger. This is a marker that's associated with your regular old, people get their cancer at 55, 60. That's the same with our marker. It's no different if you have it or if you don't. It's also, there is some variation across the subtypes of ovarian cancer. There's some thought that particularly mucinous is biologically different. And interestingly, our marker is not associated with mucinous ovarian cancer. So that's kind of consistent with being a different biological basis. Now, this is the last point, and it's fairly hot off the press and new. We had noticed that it appeared our marker for these are all ovarian cancer patients. They're people with the variant or without. And we looked at presenting disease stage or treatment response as documented. We didn't have big numbers on these. It seemed there was a trend that people with the KRAS variant did a little bit worse. But, it wasn't statistically significant. But very recently, Harvey was able to get his outcome data, his survival data on the Connecticut case control. And we combined that with survival data on 2 of our other cohorts. And actually, we're able to show quite significantly multivariate analysis just besides that, you might get ovarian cancer. If you get ovarian cancer and you have the KRAS variant, you are 50% more likely to die of that ovarian cancer versus someone right next to you that doesn't have the KRAS variant. So this is an important ovarian cancer that you really don't want to get. So in conclusion, we found this marker. It disrupts, I didn't go into too much about our data on that, but it seems to disrupt baseline levels of KRAS. Seems to be a risk for cancer onset later in life. The strongest risk is for ovarian cancer. We've found it very enriched in these families. Some of these families have lung cancer histories and some other cancers that are interesting. We're pursuing the effect on BRCA. It does seem to be a biomarker of poor response in several cancer types besides those that it puts you at risk at. And then the most important slide is to credit the people that have done all the work. Lena Chin actually made the discovery of the marker. Elena moved on with it in ovarian. These are my lab group that was very involved. Frank has been a fabulous collaborator. My department has collected hundreds and hundreds of samples, thousands almost at this point, which I appreciate. That's a lot of hard work. GYN oncology, Dr. Schwartz, Rutherford, Dr. Santine have been very involved in collecting samples over there. Dr. Elena Ratner also. Department of Epidemiology, Harvey, Herbert, Lingwen, Dan Zelterman have been really critical in the studies, pathology, Dave Tuck, genetics, Alan Matloff, and that's it. Happy to take questions.